to the cloud. Let me get in my other, here we go. All right, so we are covering the introduction to the nervous system. There's a good amount of information in it, but I really like to get the most information covered at the beginning and then kind of taper down some. We're going to do the best we can. Once you're in the nervous system, it kind of keeps flowing, because you have a lot of like a whole new system. There's a lot of terminology. And then you have the central nervous system versus the peripheral nervous system. Then you have the autonomic nervous system. And then we have special senses. Now, special senses, is a lot of structure function. You're going to combine some of the information you studied in the cranial nerves and things and apply it to special senses because, a lot of the sensory of cranial nerve from special senses is conveyed on cranial nerves. So just stay on top of it. Once you know like the basics, a little bit more than just the basics, the better off you're going to be. And there's some things that you can kind of rehearse as you go as well, especially those charts. I love those charts because they're right there. You can copy them over and over. Okay, so we're going to start with, characteristics of the nervous system. Well, compared to the endocrine system, which you're not really that, familiar with at all, we've just talked about a few hormones, is that the nervous system is very rapid, very specific, and has an immediate response. The endocrine system is a system where glands from one area of the body produce a chemical compound called a hormone, and we put it in the blood. And as the blood circulates to different organs, if you have a cell that has a receptor that hormone can bind to, then tag, you're it. I'm going to communicate with you, or it turns that cell on to do a particular thing. But the nervous system is to nerve, or like a neuromuscular junction, a nerve to another structure, a muscle cell, and so it only affects essentially what it's terminating on. Okay. So this is our jumping off point, where you can label this up with various amounts of information. We have the central nervous system versus peripheral nervous system. Central nervous system composed of the brain and spinal cord. Sensory is represented on the left-hand side, the blue information going in. I mean, it's bilateral. It's just they're comparing sensory information from signals from the external environment. The skin, tendons, and muscles, and internal organs are all the sources of sensory input, like the free nerve endings and Meissner's and Pacinian corpuscles that we looked at. We said that there are receptors for certain things, and they respond to a change in the environment, and then the impulses travel, conveyed on this kind of a cell or a wire, a sensory, to the central nervous system, where you can put meaning to it and develop a response. And then the outgoing command is in the motor division. You either have a single motor neuron to the somatic division, which is just the skeletal muscle, and then you have a 2 neuron chain. Those 2 pink arrows are part of the autonomic nervous system. And that is the motor to everything else, glands and any other smooth muscle, cardiac muscle. Right, so the somatic division, over and over again, I love that term, is going to be to skeletal muscles, right, that you've studied, and then how we control everything else. And it says in the smooth muscle, cardiac muscle, glands, that is going to be a 2 neuron chain, part of the autonomic nervous system. And that's the details of which we get into in the last chapter. So you start here by just looking at this organization. We're going to come back and be able to put in next to the sensory here. We're going to write in their unipolar neuron. Over by the motor, you're going to put a multipolar neuron, because all the motor neurons are in the shape of what's called, or category of, a multipolar neuron. These sensories are part of a unipolar. And then later, we're going to come back and maybe draw arrows from the eyes, the nose, the ears that represent special senses, quote unquote. And there you're going to draw an arrow to the central nervous system. And those particular neurons are their own category. that convey sensory information from special cells to the central nervous system. They're the bipolar neurons. We're going to come back and label these signals from external environment. They come from external receptors, signals from tendons and muscles, proprioceptors. So this is going to be kind of a jumping off part where you can label up with more detail that helps keep reinforcing sensory in, motor out, in peripheral nerves and in it's peripheral nerves, which includes spinal nerves and cranial nerves. And then you have your central processing unit, your brain and spinal cord. Okay, so principal parts. We have the central nervous system, brain and spinal cord, receives and processes and transfers information. And then the peripheral nervous system is everything essentially outside the CNS. Those are the neurons carrying information to the central nervous system called sensory neurons, and motor neurons carry information away from the central nervous system. Remember when we looked at the nervous system, we said there are two types of tissues. You have neurons and neuroglia. The neurons, these neurons, sensories and motors, are cells that convey electrical impulses. But then you have cells of neuroglia that are found in the central and peripheral nervous system, 4 in the central, 2 in the peripheral, that function in the nervous system but don't convey impulses, but they help this whole system work better. So functions, we have sensory input, information about changes in the environment to the CNS. The integration system sorts and processes, and makes an appropriate response. And then the motor output are the signals carried away on those motor neurons. Think about how a computer works, even if you don't really know a lot of it. Most of it, I think almost everybody has to take a basic, like a 1-credit kind of computer class that talks about generally how computers work. And you know that everything from your keyboard, you punch things and you type things on your keyboard are like sensory input. It sends signals to your CPU, your central processing unit. And then it sorts it, and then it sends out a signal to your monitor that displays something, about what you just typed in. And so your, computers are they stole the idea from how your nervous system works. Okay, divisions of the peripheral nervous system. We have a sensory division, transports to the central nervous system. The 3 sources are signals from the skin, tendon, and muscles called proprioceptors. Signals from internal organs, internal receptors, and signals from the external environment, external receptors. So an example of an external receptor are those receptors we find in the skin, from free nerve endings, Meissner's and Pacinian corpuscles. Internal receptors, we're going to look at more of those, or a few of those anyway, at least when we get to the cranial nerves, because we have chemoreceptors, different types of mechanoreceptors, chemoreceptors that are found in blood vessels, in certain blood vessels and within the brain, certain areas of the brain. And they monitor the chemical content, especially the amount of oxygen molecules or the concentration of oxygen, carbon dioxide, hydrogen ions, from those areas. So those are from really internal sources. So again, taking those 3 terms, now you can go back to the beginning and where you have signals from external, environment right next to it, external receptors, proprioceptors, internal receptors next to those. So you just keep, again, building and layering, on one image. Okay, all right, so we have, in addition to the sensory is carrying information to that central nervous system, we have the motor division, which is carrying impulses away, right? So motor, think about, something motor, a motor working, it means you're physically going to do something. You have a single neuron in the somatic division, and somatic just means kind of like general body to the skeletal muscles. And the autonomic nervous system is a 2-neuron chain, all right? These neurons are either going to be part of what's called the sympathetic division or the sympathetic nervous system. This is our fight or flight responses and maybe responses to immediate stress. And then we have the parasympathetic division that controls the resting and digesting normal activities. Each one, excites certain organs and inhibits others when we need to run like hell for 3 minutes, or to get away from bears that are running up behind you. And then in reverse, the parasympathetic division will stimulate the other organs and inhibit the, stimulate some organs, inhibit other organs when we just want to be in rest and digest. And what we'll find is that the organs that I excite in the sympathetic division will be inhibited in the parasympathetic division, and so forth, so that we're pushing and exciting certain organs that are associated with allowing you to protect yourself and respond physiologically very quickly to survive. And then the other organs, we're going to stimulate when we're just resting. We need to digest food and transport it. All right, so of the nerve tissue, we said we had neurons and neuroglia. We talked about that in chapter 3, when we talked about different cells and tissues. So the neurons, demonstrate electrical excitability and capable of conducting impulses called action potentials. They respond to stimuli and convert it to an impulse, or what we call an action potential. Neuroglia cells support these neurons in their functioning in the central nervous system and the peripheral nervous system. Right? So in the central nervous system, we have 4 types of cells. And these, again, make great flashcard things for you guys. Hopefully, Caitlin, you guys had gotten together forming your study group. Hopefully it worked out well, and you saw a reflection, in your exam grades. And I would keep doing it, because there's a lot of minutia in here, but there's a lot of, again, like structure function things. So there are a lot of questions come from this first chapter, because it gives you the most time to kind of mull it over. And so I would start, ASAP with developing some kind of flashcard or some kind of quizzing method on these. Like which one's in the CNS, which are in the PNS, what are their functions? Because these come back, like the brain-blood barrier, the myelin sheaths, the oligodendrocytes, the ependymal cells in AMP2. Okay, so the astrocytes. Astrocytes are cells that wrap around blood vessels and form a brain-blood barrier. They restrict the movement of material between the blood and, the brain tissue itself, as well as the cerebral spinal fluid, that passes through the around your brain and spinal cord and in the center of your spinal cord. Then we have oligodendrocytes. They produce myelin sheaths. Myelin sheaths are layers of fatty tissue that wrap around axons, in the central nervous system. In the peripheral nervous system, we'll have a cell that does similar things, but it only surrounds one axon at a time. These oligodendrocytes, they can wrap around neighboring what's called axons, or parts of neurons adjacent to them. So they can essentially do double duty. Microglia are cells that are capable of phagocytosis. They can phagocytize bacterial and microbes, as well as worn out cellular debris, cells that are dying off that need to be evacuated. And then we have ependymal cells that produce the cerebral spinal fluid, that's found in the ventricles of the brain, again, surrounding the brain and spinal cord, and in the central canal of the spinal cord. So those are the 4 cells of the central nervous system. Neuroglia means support cells. And these are their kind of shapes. You're not going to have to recognize images on a quiz or on exam in lecture, but it gives you an idea. So the astrocytes, they kind of look like a star, and they wrap around the capillaries here that contain blood, so that only certain types of molecules, can go from the blood out into the brain or into this cerebral spinal fluid. I mean, we need to deliver oxygen and nutrients. There are certain substances, certain drugs, and things that, should not be leaving the cardiovascular system. Again, certain organisms especially, but some of them can. And then over to B, we have the microglial cells. They're the phagocytic cells. They're capable of engulfing material. And then over to the lower left-hand side, we have ependymal cells. They are adjacent to them. At one side will be a blood vessel, because they're going to extract material from the plasma of blood and then produce it and secrete it into the fluid-filled cavity above it, so to speak. So that's how they're going to produce cerebral spinal fluid. And then to the lower right-hand side, we have the oligodendrocytes. The Schwann cells in the peripheral nervous system will be similar, but because in the central nervous system, we really only have neurons and maybe some blood vessels only, is that everything kind of surrounding these, or adjacent to these different nerves or just other nerves. So one cell can kind of wrap around and help insulate, their neighboring neurons and help kind of do double duty there. In the peripheral nervous system, we have Schwann cells and satellite cells. Schwann cells, like oligodendrocytes, will form myelin sheaths, but you have one cell to one discrete little area on an axon, and that's it. And it wraps around and around and around and around and around, kind of like rolling your hair up in a curler, if anybody even uses curlers anymore. And so it helps insulate, or it does insulate that axon where we have action potentials generated, and thus can help increase what's called the nerve conduction velocity, the rate at which an impulse will travel from where it originates in what's called the axon hillock till where it gets to those axon terminals where you secrete a neurotransmitter. So, when we looked at that neuromuscular junction, a lot of that is coming back. We only looked at really those axon terminals, and we had those videos how, the impulse travels and action potential travels down the axon to arrive at the axon terminals. And that stimulates these voltage gated calcium gates to open. So calcium enters the axon terminal, and that stimulates the synaptic vesicles to merge with the membrane and to release the neurotransmitter. Well, now we're coming back to all that, but we're looking at, well, what are those impulses? What is an action potential, resting membrane potential, graded potentials, where do they occur? What are these myelin sheaths? And so we're going from really those axon terminals and going kind of above it and kind of retrograde going like, well, where is that cell body? Where is it located? And so we're looking at more of the anatomy that supplied, that neuromuscular junction. Satellite cells are found around the cell bodies that regulate the exchange of materials between the cell body and the interstitial fluid. So again, they can kind of limit what kind of material enters and exit or even binds to certain receptors, on the cell body, in the peripheral nervous system. Unmyelinated axons or cell bodies appear as gray matter. So where we don't have that myelin sheath, and think, that's produced by the Schwann cells and the oligodendrocytes in the central nervous system. Think of yellow. It has it in a vanilla color. They call it white matter. But think about white and yellow being kind of synonymous because it's going to represent these fatty layers, that insulate, and kind of protect those neurons. Where we have gray matter is where you're not going to have any myelin. And generally, the peripheral nervous system, we're not going to myelate the axon terminals or the cell body because we need to be able to have an area on the cell body that another neurotransmitter from a neuron can bind to. Well, you can't cover, you don't want to cover up all the receptors because nothing can bind to it. And when the impulse travels to the axon terminals, well, the synaptic vesicles need to merge with, the axon terminal area and release the neurotransmitter. Well, if you had fat surrounding it, you'd never be able to release anything. So gray areas are unmyelinated structures. It could be some axons, especially in the autonomic nervous system. Some of those have very, very little myelin sheath, but generally they're going to be cell bodies and axon terminals. This shows you a picture of a unipolar neuron in the peripheral nervous system. It's going to be one of the sensory neurons. And here in purple, you have the satellite cells surrounding a cell body. And then, here on the axon, we have in blue, here it's represented as blue, Schwann cells that are wrap around and around and around. But you see how here, the little branching end, those axon terminals, do not have fat surrounding them. Also notice, and we're going to come back to this as well with another image, is that you have little gaps between those areas of blue, those Schwann cells that form the myelin sheath. Those little gap areas, the black line pointing to it is calling it the nerve fiber. It's going to be an axon, but it doesn't have any fat on it. So when we look at like these models of what's called multipolar neurons and sensory neurons, but that gap area is called a node of Ranvier. And this just shows you a picture, starting at the top and working your way down. You have a Schwann cell in purple, and in some of the models, it'll have like a darker reddish kind of little nucleus in there. But when you're developing embryologically and as you grow as an infant, those cells wrap around and around and around and around until finally you have multiple layers surrounding that axon. So one Schwann cell in the peripheral nervous system insulates one little area. And then next to it will be another Schwann cell with a little gap in between them, and it wraps around and around. This is going to really help nerves that are really long, that have to conduct action potentials long distances, maybe from where they're originating in your spinal cord, in your cervical or neck area. And that nerve travels all the way to some muscles in your forearm or even in your fingers. That's a long distance, so we must, put some insulation around them. So this impulse travels very, very quickly, sometimes, at a rate of about 100 meters per second. Okay, so this is, again, a lot of that structure, function, all this stuff you have to start kind of incorporating. We have parts or structures of a neuron, and our basic neuron we look at is a multipolar neuron because they're so abundant. All right, so we're just going to go through these and kind of give you the definitions, and then we can start applying them to some pictures, and especially in lab, hopefully, whether you're taking it on this campus kind of online, there should be pictures, of our models. I have some pictures of the models actually embedded, within the PowerPoint, so I can quickly, point to things. And there's some pictures from your textbook that are pretty good. All right, so dendrites. Dendrites are little processes on the cell body that conduct an impulse toward the cell body. All right, so we're going to look at those. Dendrites have receptors on them for different neurotransmitters from other neurons, in its environment. You have to picture these as being kind of like all these dominoes set up, one neuron to another neuron to another neuron, like a nerve to a muscle at a neuromuscular junction. The cell body, also referred to as the soma, is the nucleus, and the nucleolus surrounded by cytoplasm and all the other organelles you would expect to typically find, in a cell. Then you have an axon, which is a nerve fiber arising from the cell body. That is going to be the wire that kind of conducts the impulse from the cell body away from it, as long as it, is the longest distance it has to, till it finally branches off to the axon terminals, at that point where it needs to synapse with another neuron or synapse with, a muscle so it contracts, or maybe some kind of a gland. The initial segment in axon helix is kind of found between the cell body and the axon. It's this little kind of a funnel cone region, which, given the different impulses and signals from the dendrites called graded potentials, will be kind of summed up. And it's in this little area where if a certain voltage is, acquired, called a threshold voltage value, then that tells the cell, okay, I want you to send the signal, and boom, it creates an action potential, which is this kind of electrical signal that travels down the axon to the axon terminals. So it's initiated at this initial segment / axon helix. Nodes of Ranvier areas of axon where an action potential is going to occur, because it's a little naked region between myelin sheaths. So I'm going to point to that in just a minute. The axon terminal, you're already familiar with, the dilated terminal ends of a neuron. They store synaptic vesicles. You have a lot of calcium gated channels there. So when a certain voltage is acquired, then calcium floods in, and then, synaptic vesicles merge with the membrane and release the neurotransmitter. Synaptic vesicles store the neurotransmitters, and the neurotransmitters, the chemical signal, like acetylcholine, that will be released. So here, this is a good picture. It's not a model, but then when you see the model, this will make more sense. Excuse me. We have, let's start with the cell body, because that's the large yellow region with a nucleus in the center. And you can see other endoplasmic reticulum, some mitochondria and things there. And you see they kind of branch off like branches on a tree. They're called dendrites. And on dendrites, you will have receptors for neurotransmitters that are going to be released from other neurons. So right now, we're just looking at one, okay? but we're going to look at these basic structures and their functions. And now then we're going to picture, hey, if I put the axon terminal, which is over to the right side, the lower right hand side, if I took one of these whole neurons and I had the axon terminals kind of make contact or come close to the dendrites so that the nerve is then sticking off to the right or to the left, superior to it, that's how one neuron communicates with another neuron. So the axon terminals of one are going to be adjacent to the dendrites of another neuron, and they're going to form kind of a chain. Okay, so we have the dendrites, the cell body. You see that, leading up to the axon, that little triangular kind of a cone area. To me, it looks like a funnel. All right, that's your axon hillock or initial segment. Graded potentials will be developed and generated from the dendrites here, and sometimes even on the cell body. And they're going to be a type of signal that gets transported or travels to the axon hillock. And if the axon hillock is stimulated enough, so to speak, then an impulse travels down the axon. And that impulse is then what's called an action potential. So these different potentials, we're going to define those shortly. And that impulse now travels down the axon. Where you have blue areas of myelin sheath, the action potential will jump from a yellow region to another little yellow region. From the axon to, let's say, that first little yellow region between one myelin sheath to the other. And those little gaps there are called nodes of Ranvier. So over to the right-hand side, it gives you a little black line that says, hey, that's a little gap there. It's called a node of Ranvier. The impulse travels from the axon hillock, to the terminal ends, and the axon terminals where they store and release neurotransmitters. When we look at with some of these models, all right, there's one, it has the top part here, the dendrites, the cell body, and up to where you have the first myelin sheath, and it gets cut. And part of the model is just that on like, it has like two parts to this model. So one part of it is just so it's nice and big, shows you the cell body and the dendrites and the axon hillock in the beginning of an axon. And then below it, it takes the next part of that, these Schwann cells with an axon, in the center and nodes of Ranvier. And it shows you also the connective tissue that surrounds it. And this is that picture. So part of this model is, like I said, here's your cell body, the soma, a nucleus in the center. We have dendrites extending from it. Theirs are cut like little nevers because it would be too large and branching. So let me come back to A, because that's going to be the axon terminal of another neuron. But we have the cell body in the center with a nucleus. We have the little extensions D, which are the dendrites. The red pinkish part here, between the cell body and E, the axon, would be the initial segment, all right, or the axon hillock. Those are the parts you can see on here. Below it, you can just barely see, that's going to be a Schwann cell. So they cut off to the right-hand side that axon that E is pointing to, and they take the rest of, or at least a portion of what you would have further to the right-hand side, and they put it below it so that this model isn't, 12 inches high and 3 feet long. It's, like 18 inches high and, 20 inches long. So you get them kind of not superimposed, but layered on one another. These at A are axon terminals of other neurons that need to communicate to that particular neuron. So that may be multiple neurons, it may be one neuron, just depends. Now we're looking at the bottom part of that model, and as I said, shows you, okay, this is where the axon would be, which is in the center in gray. They cut it open so you can see that there are mitochondria, and other organelles. You have A, that is a little naked region of the axon called the node of Ranvier. It looks like these yellow areas, B, of myelin sheath touch there, but they don't. They come really, really close, like a neuromuscular junction, but they don't touch. All right. So the little gap, that zigzaggy region that A is pointing to, is the node of Ranvier. B is the myelin sheath that has wrapped around and around and around. And D is the Schwann cell that produces that myelin sheath. E is the endoneurium. All right, so that's the deepest layer of connective tissue that surrounds one cell, kind of like an endomysium. All right, cluster and bundle terminology. Right. So it's helpful to understand what these terms mean, because depending whether you're in the central nervous system or the peripheral nervous system, the same thing you might be referring to, such as a cluster of cell bodies or a bundle of axons, we have different terms in different places, but they may, essentially mean the same thing. In the peripheral nervous system, we call a cluster of cell bodies a ganglion. And in the central nervous system, we call them nuclei. Bundles of axons in the peripheral nervous system, we call them a nerve, because it's easy to identify them, because in the peripheral nervous system, we have a variety of different tissues. We have nerves, we have blood vessels, we have muscle, we have bone, we have glands, we have all this stuff. So when you can pick out and dissect out nerves as they're traveling. But in the central nervous system, pretty much all you have are nerves. And the only way you can distinguish them is kind of the location, maybe in the brain and the spinal cord. Sometimes the direction that the nerve by these axons are traveling will give you an indication. And sometimes they have like a slightly different staining capacity. So central nervous system, nervous anatomy, is far more difficult to distinguish and identify than in the peripheral nervous system. Right. So again, easy terminology here, work that in. If I put in a chart, you probably get at least 2 questions from it. Okay, structural diversity. So of these neurons, the neurons that can convey some kind of electrical impulse, there are 3 basic shapes, or 3 shapes, but they're found in discrete areas, which make them kind of easier. All right, so the shape of a multipolar neuron, that is the shape of all our motor neurons. And oh, look, it says, that should say interneuron. I need to make a typographical change to that. It should say interneuron, not, whatever, whatever that is, right? Motor neurons and interneurons, or association neurons, are in the shape of multipolar neurons. Right? So remember, they carry impulses away from the spinal cord. Then we have bipolar neurons. They're the sensory neurons from the special senses only, that travel, to the central nervous system. Unipolar neurons are somatic sensory neurons, which means they're from every place else in the body that isn't a sensory, a special sense organ. And, so they convey touch and stretch, kinds of vibration, pressure changes, light pressure, deep pressure, to the central nervous system. And they have kind of what we call like a hanger shape, where one end is a dendrite and the other end is the axon terminal. But I'm going to show you kind of an example. But before we do, I'm going to quickly go back here to the beginning, that first picture, and say, okay, next to the sensory input here, you see those sources, I would write unipolar neuron, because all these sensory neurons from these areas here in the blue are all in the shape of a unipolar. The motor over to the outside, right, multipolar, M&M. Motor output is all multipolar neurons. And then maybe from the eyes and nose, draw arrows to the brain and write unipolar. Bipolar there, because that's the shape of those sensory neurons that convey special senses, to the brain. You can do that, not do that. I just like to embellish like one big picture, so it works to have multiple features, and instead of going back and forth to too many different PowerPoints. Okay, so now we're going to look at these shapes. So that model that we looked at, we call it the Lisa Simpson model, because it kind of looks like Lisa Simpson, that the little braids and things are extending from her head. The multipolar neurons convey motor impulses away from the central nervous system, most abundant in the major neuron in the central nervous system, and in the peripheral nervous system, really. Bipolars are rare. They're found in special sense organs, and they convey special sense information, from the receptors, the chemoreceptors in your nose, the photoreceptors of your eyes, hearing in your ears. Right? And then the unipolars are found mainly in the peripheral nervous system because they're conveying sensory information from all those external receptors, in the peripheral body, and they have a certain shape. The bipolars are very linear, they're continuous. They go from dendrite to cell body to axon, very linear. But look at the unipolars. If you could see the end of that axon, which is, to the inferior aspect here, it's going to kind of branch off the same way as the receptor endings, to the superior aspect do. So we say it kind of looks like a hanger, where the cell body has a little neck there. And if you could, you can put a shirt on there and hang it up in your closet. That is the shape of a unipolar. Right, so unipolar is the sensory portion, that blue areas on that first diagram we looked at. The multipolars are all the motors going out, either to the somatic, to the skeletal muscle, or to the glands and parts of the autonomic nervous system. And bipolars are conveying sensory, from special senses, from those organs to the central nervous system. Okay, so let's just go briefly, we're going to define a synapse. A synapse is like a neuromuscular junction. It's just where one neuron meets with another. And so we call it a synapse because we're trying to say that it's not a nerve to muscle junction. This is a nerve to nerve junction, but it has the same features of having a cleft or a gap between them. And because impulses travel in a singular direction from where the impulse is generated by the cell body toward the axon terminals away from the cell body, then the impulse is carrying the first, or the neuron that's carrying the initial impulse is called the presynaptic neuron. Then you have the gap between them, thus the synapse. And then you have the cell on the other side, the postsynaptic neuron. So, on that model that we looked at, they looked like little chocolate kisses that were on those dendrites. That's what we're trying to represent here, especially this one up at the top that shows a neuron, with an axon terminal and that black circle around it. That's what they're trying to represent there, and further, that black circle is illuminated up in the yellow up there. That's the synapse from one neuron in its axon terminal to the dendrite, or maybe even cell body in the case of the neuron in the bottom part of that image. So, it has little synaptic vesicles with little blue dots in it, and it's being stimulated. So it's releasing the neurotransmitter to the gap, and it will bind to a receptor, on the postsynaptic membrane. And the same things happen to it as in a neuromuscular junction. It can be broken down by an enzyme. It can diffuse away. You might have reuptake. The same, the fate of those neurotransmitters are the same at a neuromuscular junction, and that is synapse. Okay, then we move on to these different signals. Right? And electrical signals in neurons involve ion channels and membrane potentials. Membrane potentials, we have three types: a resting, a graded, and an action potential. And we're going to define those in just a minute. Right? But we also have to identify what these different channels or gates are that we find on the surface of these neurons on their plasma membranes. So we have four basic types. We have leakage channels that randomly open and close. And generally, all these channels, if they allow ions to pass through them, or in the case of a ligand at number 3, or what we call a chemical channel, maybe, it's going to be to a particular and single hormone or neurotransmitter. And the other channels that allow ions to pass through them generally are for one ion, either sodium, calcium, potassium. Okay, keep that in mind. Okay, so leakage channels just kind of randomly open and close. Ding, 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 ding. And thus it will be the reason why we're using our sodium potassium pump kind of regularly. Voltage channels open when a particular voltage value or charge is acquired on the membrane. That's going to be required for those calcium gates to open in the axon terminals, and for sodium potassium gates to open and transmit what's called, either a greater potential and or more importantly, an action potential. Ligand channels open because a particular hormone or neurotransmitter bound to it. And then mechanical gates or channels open because some kind of physical vibration, pressure, or stretch. So, those, remember the Meissners and Pacini corpuscles, they responded to light pressure and deep pressure. They're a type of mechanical receptor. They, are stimulated because something physically happened. You squished it, you bent it, you did something. That's what we call being mechanical. Let's think of it as something physically moving. But the rest of these, either the leakage kind of randomly open and close, the voltage, a particular value has to be obtained for a sodium gate and a potassium gate or a calcium gate. And then the ligands open up because a particular chemical compound of some kind bound to it. That's why they can also be referred to as a chemical channel or gate. And therefore, right, certain ones are going to be found in higher populations on certain parts or areas of that neuron. All right, so we're going to review. We talked about this, a little bit when we looked at cells, but we need to know what are our potential diffusional pathways and where, for these different ions when these gates open up. So forever and forever, right, potassium ions are always the cation that is in highest concentration inside the cell and very low outside the cell and more importantly in plasma, in blood, right? It should be very high in the cell, but very low in plasma, right? It should only be about 3.5 to 5.0 milligrams per deciliter, something like that. And sodium, along with chloride, should be in higher concentration outside the cell versus inside the cell, always, always. Therefore, we have this potential for diffusion, because remember, diffusion is diffusion really of everything other than water for all intents and purposes from an area of high concentration to low. So, if any of these channels or gates that open, let's say a potassium gate opens up or a sodium gate opens up, we can predict which direction they're going to travel from inside the cell to outside the cell or outside the cell to inside the cell. But we need to keep them separated like that. It's like right now, we want to keep cold air inside your house or in your car and hot air outside. And that takes energy, right? Because you have to keep pumping, you have to cool air and pump it into whatever room or in your car that you're at, shouldn't say where you are. And in the case of a cell, it maintains that distribution, unequal distribution of ions, using the sodium potassium pump. Ding, 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 massive ding, ding. It pumps 3 sodium out and then grabs 2 potassium in and brings it into the cell. 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. We'll talk about, what happens with these action potentials. And what I want you to keep in mind is that right now we're pretty much talking about these nerves, and this distribution of ions is the same in muscle cells and in especially cardiac muscle. And the directions that they're going to move, will either help you have an impulse for your heart to beat, or it will not. It can either overexcite it and make your heart beat too frequently, or underexcite it and not let any contraction occur, in which case you're in a lot of trouble, right? So potassium, a very, very, very important cation to keep in mind going forward throughout your education, because those potassium ions in the wrong place at the wrong time, can send your patient into a lot of cardiac instability, if not death. And that's why, when those values change, those are criticals, those have to be sent to the physician and, responded to very, very quickly. Okay, love this little picture. This is going to be double duty also. All right, so here we have, a multipolar neuron, right? Right, so you have the cell body and the dendrites, they're covered in blue, right? And the blue area represents, this is the part, the plasma membrane includes chemically gated channels or ligand channels. So you could put chemical or you can write ligand next to it, because that is where we have all these receptors for different neurotransmitters where another nerve will probably be communicating with it, like we saw in that other image. Right? So that is where we have chemically or ligand channels for hormones and neurotransmitters. Right? We stimulate it enough, and then at that little cone area, as it meets the axon in green here, we create an action potential if it's significant enough. And now the impulse travels down the green covered axon to the axon terminals. And that green region is telling you in this area, we have voltage gated sodium and potassium channels. They're not going to be ligand, they're not chemically gated, they're all voltage gated. And then at the terminal ends in the little toes, the axon terminals here, we have a red area, and those are voltage gated, but they're just for the calcium. So remember when we looked at those videos, you maybe go back and look at that muscle video and you go, remember they were talking about impulse travels down the axon, until it arrives at the axon terminals. Voltage gated channels open and calcium permeates into the cell. It binds with the synaptic vesicles. See, this is what they're talking about, that those particular channels are voltage gated for calcium so that it stimulates the release of your neurotransmitter. We're actually going to come back here again after we cover the potentials. And in this blue area, I'll just tell you, you could write in there right now, graded potentials, because that's where we produce graded potentials. And along the green and red will all be action potential territory. So, these images can communicate more than one thing. All right, so we're going to define what these potentials are. You have resting membrane potentials, action potentials, and graded potentials. But first we have to know, well, when you say potential, what are we talking about here? A potential, what they're talking about here, is the charge across the membrane of a neuron. What is the difference in positive, negative ions and amino acid charged entities? What's the difference between the immediate inside of the plasma membrane, or the neural element to the outside? Right? So that what is the charge distribution, and the resting membrane potential, right, is in these cells is generally negative 70 millivolts. Right? So what we mean by resting membrane potential is these neurons, all right, their only job, like we said before, is to send an impulse or some kind of a signal. They're not always at work. It's like turning on the light switch, turning on the switch to send a current to your light bulbs. When it's off and it's at rest, right? So when a cell is not doing anything and it's hanging out, waiting to maybe send a signal or not, we call it the resting membrane potential because it's essentially at rest. Right? And the charge distribution, if we measure it with a little voltage meter, and we're going to give you an image of that, the interior should be negative compared to the exterior. Some of these videos will review that. And the resting membrane potential of RMP should be about negative 70 millivolts in these neurons, and in most of the muscle. In cardiac muscle, it's a little different. It's not going to be quite, negative 70. And it's maintained by the sodium potassium pump. All right. So here, we have an axon. They're saying, hey, take that little region of the axon. If we can measure the voltage between the inside and the outside of that axon, the meter would read about negative 70 millivolts. All right, the inside should be negative. The outside immediate to that membrane should be positive, right? At rest, not when it's active. Okay. So the resting membrane potential averages negative 70 millivolts. It's the voltage difference between the inside and the outside of a living cell, and when it's at rest, it's the resting membrane potential. And it's said to polarize the membrane because it's like a polar covalent bond. One side is negative, one side is positive. All right, that's polarity. Potential energy exists due to the difference in the ion concentration of sodium and potassium. Right? Potassium is the highest, should be highest inside the cell. Sodium is highest outside the cell. Remember, sodium and chloride, where salt goes, water goes. When we looked at tonicities, and their effect on cells, we said, what keeps more fluid outside the cell than rushing into the cell is that you have so much sodium chloride out there, and where sodium goes, chloride goes, and where salt goes, water goes. So there's no way that you'd have a high sodium, chloride um, content in the cell because it would draw so much water osmotically to it that the cell would blow up and rupture. So that's one way to kind of keep in mind sodium chloride outside the cell, potassium high concentration inside the cell. All right, so oopsie doodle. Then we go on to now graded potentials and action potentials. All right, so this is another, again, flashcard material. If you know the characteristics and features that distinguish a graded potential versus an action potential, this is so much easier. And you just keep repeating it over and over. These, again, make great little questions for quizzes and exams because, you have now like 2 weeks to practice this stuff. Right? Okay, so a graded membrane potential. Right, now something is happening. It's not at rest anymore. Graded potentials are the kinds of potentials that occur on dendrites or even the cell body. So back at that little picture before, the blue areas that represented the chemical or ligand-gated areas or channels. That's what we're talking about graded membrane potential area. Their potentials, the effect that they can have on that membrane, starting at negative 70 millivolts, it may, cause the interior of that cell to change dramatically. And it may cause the interior to come either more positive, getting closer to 0, like going from negative 70 to negative 60 or negative 40, or it may make the interior to become more negative, hyperpolarized, it's called, going from negative 70 to maybe negative 80 or negative 90. These potentials can have variability, right, because they're being produced by different neurotransmitters binding to these receptors. So that's another characteristic. They can vary in size and strength or amplitude. And they only have to travel short distances. They just travel from where they're generated on a dendrite maybe to the axon hillock, very short little distance. Action potentials are different. Action potentials are the signals that occur on the axon. They are produced and originate at the initial segment or the axon hillock. They travel on the axon alone. Right? They will produce one wave, and the same size all the time and the same shape of it. Kind of looks like an EKG wave. Right? It will never vary normally. Right? It will never ever vary. And it will go from a threshold point of generally negative 55 millivolts to plus 30 millivolts, and then back down to being negative again. It's like turning the light switch on and off. They travel long distances. Those axons, as I was trying to explain before, sometimes they're very short. They maybe travel from, let's say, the neck area to some of the immediate muscles in the neck area, let's say to levator scapulae or something, but sometimes they have to travel all the way to your fingertips, or the neurons, the motor neurons that leave your lumbar spine and travel to the muscles in your lower extremities, down way into your legs and into your feet. Those are very long distances. Right. So action potentials travel long distances, not short distances, and thus the reason why we want to myelinate them so they travel very quickly. And they are an all or nothing principle, means that once the threshold point to generate an action potential, which is negative 55 millivolts in almost all these cells, then it happens without any choice. It's like having all these dominoes lined up. Once you push over that first domino, as long as they're spaced appropriately, all the dominoes will fall until you get to the end, and it won't stop. So that's an action potential. It's designed, to travel all the way to the end, and then we're going to put the dominoes quickly back up again by letting potassium being flow out of the cell, and now it's ready for another action potential. So I would commit to memory these features. That's what, if we go back to this other picture, I was saying the blue area here is graded potential territory, where we have these chemical or ligand gates on the blue areas there. That is where graded potentials are generated. In the green and red areas, that is where we have action potentials. So now, what we're doing here is we're looking at a graph of the changes in the voltage across the membrane. We're looking at a cell, and we have, let's say, at the axon hillock, we're kind of looking at what kind of impulses are being generated, from one nerve to another, and is it significant enough change to get that, stimulate that neuron to send a signal, an action potential down its axon, or is it not significant enough? Because not all release of neurotransmitters onto the dendrites of another neuron is going to stimulate an impulse. So if we look at this graph, which is a really good one to label over and over again, and I believe I do have something on there, that will that I labeled up and I used last semester, that we'll use one image to label up what gates are opening and closing, and what are excitatory and inhibitory postsynaptic potentials and things. Essentially, it's a picture like this, but with more detail added to it. And I believe it's up there, because I used it last semester. But let's look at what this is trying to tell you, because you're going to get things like this in cardiac muscle, like the second chapter in AMP2, because this is how impulses and how we generate impulses for cardiac muscle contraction. But it's going to be very similar to this, but with what we call a calcium plateau. And they're going to say, well, remember all those action potentials you looked at in AMP1? And you're going to go, huh? Well, this is what we're referring to. Okay, so on the vertical axis over on the left-hand side, we have the membrane potential, like the little voltage meter is measuring in millivolts, right? And on the horizontal, we're just starting the clock, and kind of like in doing when you take recording an EKG, picture the paper moving under a needle, and that needle will go up and down and, draw a line. All right, so we just start the clock at 0, and you let the tape kind of run, and that voltage meter is telling you, okay, here we are at -70 millivolts. And we have time in milliseconds. You get to 20 milliseconds, nothing's happening, but then you get to about 35, I guess, millivolts, and you have that little black arrow there. And that's telling you something, something is happening. There's some kind of a stimulus. So the flat line there prior to that is your resting membrane potential, thus labeled resting membrane potential. But then you have this upward deflection, kind of a positive deflection from -70 to almost -60, but then it comes back down to resting membrane potential. That little upward deflection that doesn't reach threshold, which is represented by the dotted line there on the horizontal, means that is some kind of graded potential. All right, it went up a little bit, and then because we didn't have another stimulus, which is the next black arrow, just like in muscle, if you just have that one twitch, that one signal, you have all the events of one muscle contraction, and if you don't have another one, then we essentially put everything back. You put the calcium back in the sarcoplasmic reticulum, myosin lets go of the actin, troponin rolls back, we degrade the acetylcholine. We put everything back in the event for another impulse. So here they're trying to indicate to you, it's very similar. If you have one little impulse, yes, it makes the interior of the cell more positive, called depolarizing it, but it never reaches that dotted line. That's the threshold. That's the point where we generate an action potential. And then the sodium potassium pump gets everything back to RMP, and you have another signal. And now that other signal at about 45 milliseconds causes the interior to become more negative. When you become more negative than you started, than your resting membrane potential, it's called being hyperpolarized. It gets you farther away from threshold. Those little upward deflections are called excitatory postsynaptic potentials. So we can come back later and label that up. And the little negative ones, those little hyperpolarizing ones, are called inhibitory postsynaptic potentials, or IPSPs. All right, so then we move along, we get to 60 milliseconds, and now about 65 milliseconds, look, now we have two black arrows together. That was like two impulses arriving on a muscle cell. If we have them close together, they wave summate, they add together. right? The closer together they are, the less opportunity that sodium potassium pump has to put everything back. And so they add together a summate. But even still, it still did not reach, although it was a depolarizing potential, but it was a summarizing depolarizing potential. It still never reached that little dotted line. Right? So again, nothing happened, at the axon hillock. But then we get to about 90 milliseconds. And look, now we have three stimuli, boom, 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 and they add together. And as a consequence, the interior of the cell changes from negative 70 millivolts to about negative 55 millivolts. Over on the left-hand side, they have negative 50. It's negative 55. I'm just telling you, and everything going forward will tell you negative 55. But you'll never have a question that tells you that, asks you if the resting membrane potential is negative 50 or negative 55. It'll be negative 55, negative 40, negative 65. I mean, it'll be obvious. It's the only answer. Okay, but once you hit threshold at the axon hillock, now we generate the action potential that's now traveling down the axon. And that produces the same shape of a wave here called the action potential. The upward deflection, which will really go to about plus 30 millivolts, that's another error of theirs. And then the downward deflection, so the interior of the cell becomes very positive, then very negative, and then back to resting membrane potential. Right? That is an action potential. And we're going to look at what gates are opening, closing, what direction are these ions moving, right? And that will explain how the interior of the cell became more positive and then how it becomes more negative. And I'll tell you that an action potential, it will always become the same positive charge to the hyperpolarized negative charge and then back to resting potential. So that wave will not ever vary. It won't go upside down normally. It will be the same wave all the time. And that's what we mean by kind of an all or nothing principle with no degradation of it. It's the same shape and size and amplitude of the wave all the way down the axon till it gets to those, to the axon terminals. All right. So impulse generation, these terms, and what do they mean? Depolarization means a decrease in the membrane potential, which means you're getting closer to zero, closer to threshold, less negative. All right. Hyperpolarization means you're increasing the membrane potential, or you're becoming really more negative than you started, more negative than your resting membrane potential. Gets you farther away from threshold. And threshold, generally is negative 55 millivolts. It's the threshold is the voltage value for sodium and potassium gates to open up on the axon, essentially. And thus, that is the beginning of our action potential. Right? They are all or nothing principle. It means that the action potential either happens completely or it doesn't happen at all. So again, I got to go back to a light switch. When you put pressure on the light switch, let's say to turn it on, and it's in a down position or inferior position, if you just put a little bit of pressure on it and it doesn't flick upward, and it kind of moves a little upward but comes back down, and you just put a little bit of pressure, you move it a little bit, and then you release the pressure and it falls back down. That's sort of what a graded potential is like. But once you put enough graded potentials at the axon hillock where you put enough pressure on that light switch that it toggles kind of upward and it flicks on, what happens? Well, electricity then travels along the wires to your light bulb, and the light bulb illuminates, right? That is what an action potential is like. Once you flick that switch, it happens without, any interference normally, all right? And that's why it produces the same wave the whole time, or down the entire length of the axon. All right, graded potentials can be, as I mentioned before, excitatory or inhibitory. EPSPs, excitatory postsynaptic potentials, get it closer to excitation or threshold. So they're those little upward deflections, little depolarizing deflections. And inhibitory postsynaptic potentials cause the interior of the cell to hyperpolarize, get it farther away from every meeting threshold. Removal of neurotransmitters are by the same means. Diffusion, an enzyme maybe. In the case of acetylcholine, it would be acetylcholinesterase, or maybe reuptake at the axon terminals. right? And this just kind of shows you, hey, this is an EPSP versus an IPSP. The little upward deflection, excitatory postsynaptic potential. A downward deflection, making the interior more negative, that's an inhibitory postsynaptic potential. Right, and that takes us to our first video. Hopefully this is going to work right. Everybody can see that, right?
The human brain alone contains
Okay.
about
Thanks,
100
Rachel.
billion nerve cells called neurons. A neuron, like every other cell, has positively and negatively charged ions inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of minus 70 millivolts. The resting membrane potential depends on two factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at minus 90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to minus 70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
And of interest, when it comes to cardiac muscle, in cardiac muscle, it even has that many, even more, potassium leakage channels, and so its resting membrane potential is more negative than what I call the average bear, but than these neurons. All right. So again, this is just kind of showing you again on a dendrite, when we're talking about changes in these depolarization versus hyperpolarizing potentials. I think I could probably get rid of that one because it's redundant to the others, but anyway. Okay, so chemical synapses, are mostly what we are going to be looking at. The axon terminals on presynaptic neurons contain synaptic vesicles, which contain the neurotransmitters. Receptors are on the postsynaptic neuron. And the gap area, the region between them is called the synaptic cleft or gap. It's just a fluid-filled space that, connects one neuron to another. And thus, because they don't touch, you have an electrical impulse on one cell, then it has to use a chemical signal, and that gets converted to another electrical signal in the next neuron. It's like what happens at a neuromuscular junction. Conduction across the chemical synapse. Remember, action potentials cause calcium voltage gates to open in the axon terminal of the presynaptic neuron. Calcium enters the presynaptic neuron and triggers exocytosis of the synaptic vesicles to merge with the presynaptic membrane and release the neurotransmitter. Neurotransmitter binds to the receptors on the next cell, and it binds to other chemically ligand-gated ion channels open in the postsynaptic membrane. A type of ion channel open determines if a postsynaptic cell is going to be an IPSP or an EPSP. So again, it's just going to stimulate those open up certain chemical or ligand gates on the dendrites or cell body of the next neuron. And depending on what the chemical is, and it's not always acetylcholine, we have many different types of neurotransmitters, that are utilized in the body, to communicate. Right, so that takes us to a chemical synapse.
We have examined the anatomy of a synapse. Now, let's look at the details of synaptic transmission. An action potential in the axon terminal causes voltage-gated calcium channels to open and calcium to enter the terminal.
Professor, we can't see the video.
Calcium
You can't,
channel
okay, hold on.
anatomy of a synapse.
Let me turn that off because sometimes it, excuse me, it shares it and sometimes it doesn't. All right, that's why sometimes I'll ask, you can see this, right? Because I don't know. All right, so let me do this. Now, can you see it?
Yes.
Okay, here we go.
Now, let's look at the details of synaptic transmission. An action potential in the axon terminal causes voltage-gated calcium channels to open and calcium to enter the terminal. Now, let's watch the calcium channel in action. The presence of calcium inside the cell causes the synaptic vesicles to fuse with the membrane. Each vesicle releases a fixed amount of neurotransmitter into the synaptic cleft. Neurotransmitter diffuses across the synaptic cleft. Neurotransmitter binds to a receptor on the postsynaptic neuron, where it can act directly or indirectly. Chemically gated ion channels remain open as long as the neurotransmitter is bound to the receptor and are not sensitive to changes in the membrane potential. Synaptic current, or ion movement, through chemically gated channels may depolarize or hyperpolarize the neuron. This example illustrates depolarization of the postsynaptic neuron. Synaptic transmission ends when the neurotransmitter dissociates from the receptor and is removed from the synaptic cleft. Most often, the neurotransmitter is pumped back into the presynaptic terminal or into nearby glial cells. Here we illustrate the neurotransmitter glutamate being pumped back into the presynaptic terminal. In some cases, the neurotransmitter is broken down by enzymes, and the breakdown products are taken up by the presynaptic cell. The neurotransmitter acetylcholine is an example of this process. When breakdown products are transported into the presynaptic terminal, they are used to resynthesize neurotransmitter. The neurotransmitter, which is being returned to the terminal, is repackaged into vesicles for storage and subsequent release. The mechanism by which neurotransmitter is returned to the terminal is specific for each neurotransmitter and can be selectively affected by drugs.
Okay, so I think that's kind of how, it's a little lengthy. They could have sped that up a little bit, but okay. So let me stop that share and get back to my notes, which is this one. Okay. Okay, so generation of the action potential, we start at the resting membrane potential, voltage gated channels for sodium and potassium are closed. All right, so now we're going to be looking at, so once we hit that threshold point, like what is opening and closing, what direction are these ions moving? So the first upward deflection part of the wave of an action potential, not the graded potential. Now we've met threshold. We want to look at the action potential. Right? That upward deflection is, you're going to label it depolarization. Graded potentials increase the permeability by opening sodium gated channels. So sodium gates open widely at threshold, and sodium, and you write on next one, sodium enters the cell, and you label that upward deflection depolarization. When depolarization reaches a critical level called threshold, sodium voltage gates open, sodium rushes in the cell, generating an action potential of the axon helix. So that's why the interior is becoming more positive, because sodium is going from outside to inside, and sodium has a positive charge. The action potential becomes self-generated as sodium enters the neuron through voltage gates and causes other sodium gates adjacent to it to open. And so what you have to kind of picture is that as sodium is entering at one point, those positive charged sodium ions get kind of attracted, because opposites attract, to the adjacent membrane where it still has a negative charge. And in doing so, the sodium gate's kind of drawn to those negative charges. And now that negative 70 millivolt membrane potential at that point starts to become depolarized and get to negative 60. And then if it reaches negative 55, now the sodium gates right there are going to pop open and even more sodium floods in. And so again, it's kind of like knocking down one domino to another, that these sodium gates along the length of the axon are going to start popping open, and you're going to get sodium, rushing into the cell. But as quickly as they fell down, we start putting them back up again, because potassium gates will open. Sodium gates open and close very rapidly. Potassium gates open really slow and close really slow, like a big old drawbridge. Okay, so once the sodium gates close, right, we no longer become positive in the cell, and that's where we reach the peak of that action potential, and it stops at about plus 30 millivolts. Now, right, the downward deflection, which we're going to label repolarization, now finally the potassium gate opens. So you're right on the peak of that wave. Potassium gate opens, and now potassium leaves the cell. And thus, because you're losing positively charged ions, now the inside is becoming negative again. And it continues to become negative and even go beyond your resting membrane potential and hyperpolarizing it, because the potassium gates close so slow. And then finally, boom, they close. Well, now I'm at like negative 75 or negative 80 millivolts, just whatever value you want to choose. And finally, at the bottom of that wave, now potassium gate closes. Right. So how do we go from negative 80, let's say, back to resting membrane potential at negative 70? The sodium potassium pump. All right. So when you're looking at these videos, look at the ball colors. Right. Sodiums are generally in the red and the potassium's in the blue. At one point in this video, it says, it's that the leakage gates get you back to resting membrane potential, and that's not the case, and it's not what your textbook says either. So sometimes in their videos, the publisher and the rep from the publisher says that they get the videos from somebody else, that's supposed to be illustrating, what is being taught in that chapter, but sometimes they don't match up. So maybe they need to get new videos, but in any event, perhaps now they've updated them. I don't know, but it's the sodium potassium pump that gets you to the resting membrane potential. So and we have this basic action potential. All right, so one is we're at resting membrane potential. We have graded potentials that occur somewhere and get that nice purple line, to meet threshold. And then what do we have? We have the same wave. We have the upward deflection called depolarization number 2 to about plus 30 millivolts. It stops. Now, we have repolarization, the downward deflection at 3 until that potassium gate closes at 4. We're now hyperpolarized. And then the sodium potassium pump gets you to number 1 again. So where 2 is that upward deflection is depolarization. At the dotted line there at negative 55 millivolts, on that side of the depolarization wave, I write sodium gate opens, and then at the top of that wave, sodium gate closes. And then next to depolarization, right, sodium enters cell, because you need to know what direction these ions are flowing. And then after the sodium gate closes, and now the purple line is going downward because you're repolarizing, I would write there at the peak, potassium gate opens, label repolarization, and then write potassium out of the cell. And then at the bottom there, where number 4 is where we hyperpolarize, I write potassium gate closes. And I might, kind of just put a little bracket between the negative 70 millivolts and the value you're ending at, that hyperpolarized value. That is your area, that's the hyperpolarization. The difference between the negative 70 millivolts and the ultimate negative value you end at when that potassium gate finally closes. And then we go from 4 back to 1. That is the sodium potassium pump, 3 sodium out, 2 potassium in. So I have like a handwritten version of this labeling all that, I believe on the course page. So when we're done, I'll just share that, and I'll just go in and confirm that it's still there. Okay. All right, this is showing you here we have a multipolar neuron up at the top. We have its little dendrites, and the green represents the axon terminal of another neuron. All right, so we're looking at these synapses, right? So the first one is showing there's no summation occurring. All right, we have an impulse traveling down that green axon to the dendrite in the upper left-hand corner. All right, and down on the graph there, it has the red arrow. It says, okay, here's one impulse on that neuron, and then here's another impulse, and they're spread apart so much that one little impulse is not enough to reach that negative 55 millivolts, the threshold point to generate an action potential. So what happens? Your sodium potassium pump helps you get right back to resting membrane potential at negative 70. And you have another stimulus, but like that other the diagram I showed you with the black arrows, is that both of them are still of equal size and amplitude. They're never reaching, threshold point at the axon helix. So we don't generate an action potential. They're too far apart and they're not summating. The next one where we have temporal summation, B, you have, okay, now on that green up on the top there, look at that green axon, right? We have 2 impulses traveling close together, and thus it's reflected here in the graph. 2 of them summate, and look, when they happen together, it causes the membrane to reach threshold, and you have depolarization, repolarization, hyperpolarization, back to resting membrane potential. Your action potential wave will always look the same. All right, so that's one way we can generate an action potential. Or we can have what's called spatial summation. And look at the neuron above that here on these dendrites. One neuron on one side of the cell is exciting it, and one on the other side of the cell. It doesn't have to be the same neuron. And together, again, by the time those charges arrive at the axon terminal, at the axon helix, they summate. It's just the difference between, I'm not going to ask you the difference between a temporal versus a spatial summation. We're just saying it can come from 2 totally different neurons sending signals that arrive at the same time virtually, and they will get added together and cause an action potential. Or lastly, over on the right-hand side, we have the action of 2 opposing neurons, one sending an excitatory message, the other down below in red is sending an inhibitory message. And what happens at the axon helix? Well, they just kind of cancel each other out. You never reach threshold. And so these are the different combinations of types of excitatory and inhibitory postsynaptic potentials, and whether, hey, depends on how many you're communicating to that one neuron. You can have one inhibiting it, but 3 others exciting it, and they're going to essentially, the excitation is going to cause that membrane potential to become positive enough to create an action potential. Okay, so that takes us to one of the last videos, and this is the one where I believe it says use voltage gate channels to get back to resting membrane potential. It is not. It is a sodium potassium pump.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold.
I'm just going to stop this. Excuse me, really quick. Remember, she's saying voltage gated channels. So now you know I must be on the axon, right?
Stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts.
I'm just going to stop quick here again. So again, like there's a lot of, they're trying to code this visually. The pink, are your graded potentials. And once you hit threshold and you're generating action potential and it's in sodium movement, now they switch to the red color again.
Voltage-gated sodium channels close and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, so now we're going to talk to, and by the way, so when you get to cardiac muscle, you're going to look at the action potentials of cardiac muscle. And it's going to be very similar. You're going to have repolarization. And when you start to, depolarization first. And when you enter into repolarization, we're going to have an extra phase. We're going to have what's called a calcium plateau. Yes, the potassium gates are going to open up just like they, you would expect them to. But you're also going to have calcium gates. Right? So as the potassium gates finally open, a calcium gate is going to open. And as you lose potassium, calcium enters. And that will cause a relatively flat line. It's called a plateau. It's not completely level. It is still declining a little bit. But, we have, as you lose a potassium that's positively charged, you acquire a calcium that's positively charged. And the voltage across it kind of stays rather stable for a while until that calcium gate closes. And now you complete the repolarization, hyperpolarization phase, because now the only ion moving is potassium, and you bleed out the potassium, and it allows the interior of the cell to become more negative. That calcium plateau is what allows cardiac muscle to contract and hold and squeeze and then relax. Right, so you're not sending multiple signals like in skeletal muscle to contract and contract and contract. How cardiac muscle works is a little bit different, and you don't want it to just contract and twitch. You want it to contract and squeeze and be able to push the volume of blood in a chamber from one chamber to another, or from a chamber into a blood vessel, pushing it forward forcefully. And so you're going to come back to that action potential and address what's called the calcium plateau. Thus, this is why these potassium levels are very critical. You need to have more potassium inside the cell than outside the cell. Potassium levels changing in blood plasma affect now how cardiac muscle works. It's going to change that action potential, right? Whether you're meeting threshold or not. Okay, so now we're going to look at impulses and how they are transported, or how they're generated and passed down the axon. Because conduction of nerve impulses or action potentials can either be what's called continuous or saltatory. Continuous conduction, we have the same depolarization, repolarization, hyperpolarization. That wave is going to occur down the entire length of an axon that doesn't have any myelin. Right? There's no myelin. There's no areas of fat cushioning it. So this process has to occur all the way down the length, meticulously down the length of the axon until it reaches the axon terminal. It's a very slow method. So they're going to show you what this looks like. That only travels at like 2 meters per second or something. But if we have those Schwann cells producing the myelinations, the myelinated regions, then this whole process of depolarization, this action potential, only has to occur at these naked regions. And thus it looks to kind of skip from node of Ranvier to node of Ranvier, and kind of skip right like along the length of the axon until it arrives at the axon terminals. And thus, the amount of time it takes to travel from where the action potential is generated at the axon helix to where it arrives at the axon terminals, is hippy skippy fast. I mean, it travels sometimes around 100 meters per second. So that's what you want. You want myelinated fibers, to these nerves that are going to your muscles to get them to contract, so you can remove your hand from a hot surface, let's say. All right. So that will be the subject of the next video. Right. Propagation of an axon terminal is similar to a domino effect. Change in one area affects an adjacent area. So it's going to show you how these sodium, ions enter. And as I said, they kind of get attracted to the negative end adjacent to it. And it helps that area at least reach the threshold point, because it's becoming more positive, and then that sodium gate opens. Right? And picture a bunch of dominoes. When sodium gates open, all the dominoes start to fall down. As the potassium gates open, it's starting to reset the voltage in the interior and get it ready for another impulse. So as the dominoes are falling down, you go back to the beginning and you quick start putting them back up again. So that again, if we have enough graded potentials to help you reach threshold, then again, you can knock down that first domino and they all start to fall back down again. That would be a subsequent impulse. Right? Not all graded potentials become action potentials. You must reach threshold. During AP generation and a short time afterwards, the neuron cannot respond to another stimulus. That's called a refractory period. All these things have to be kind of put back, get those dominoes back up again, and then there's a certain point where, okay, we can stimulate it again and start knocking those dominoes back down again. And thus it allows and ensures that the action potential in the signal travels in a single forward direction and doesn't back up and go like partway down the axon and then head back toward the axon terminal. It'll never, the axon helix, it'll never happen. It will always go in a forward direction.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay, so that's what we mean, the difference between continuous. Continuous is all that actually has to occur all the way down the length of the axon versus saltatory conduction, it travels very, very rapidly. I love those pictures because when I took these initially, you just had to imagine what happened and you didn't have any videos at all. All right, conduction velocity also depends on these two factors, and part of it is that myelin, right? One is axon diameter. The larger the diameter of an axon, the faster it can transmit an impulse. And then the other variable is degree of myelination. The more myelin you have, the more it increases the nerve conduction velocity. You will have, it will then, because of the presence of myelin, you will have saltatory conduction. Right? So those two factors, for once, it's good to be nice and wide and fat on the surface because you will increase your nerve conduction velocity. You will be demonstrating saltatory conduction, or you can have no myelin, right? Thus, you have a slower nerve conduction velocity because it will demonstrate continuous conduction. All right, so axon diameter, myelin sheath. The bigger the diameter, the more myelin, the faster you transmit. You decrease the diameter, you decrease the fat surrounding it, the slower your conduction velocity. All right, and then again, phytoneurotransmitter, diffusion, enzyme degradation, reuptake. Any of these three can happen. The video doesn't really talk about any kind of diffusion, but it occurs. And that's it. I know this is a rather, let me just stop my share. I know it's a long chapter. I mean, and I get it, but it's a double lecture. And generally, if we were face to face, we'd take a week to cover this. I'd usually cover all the anatomy and then cover all the action potential stuff in another lecture. But, this is your foundation, for this whole chapter. And then the rest of it is, and I think this is the hardest part. And once you get the difference, I mean, between all these different potentials and the different gates for things, it carries over, later, to special senses and then further into AMP2. So that is the end of the lecture. Let me just stop recording here. Okay, do you want to stop recording?